Okay, so now we can go on to the organic molecules. So organic molecules make up a large portion of our body. We also have the inorganic molecules in our body, but I want you to know the difference. What an organic molecule really means is that if the molecule contains both carbons and hydrogens. Okay, so glucose, for example, its formula is C6H12O6. Because it has both carbons and hydrogens, it's organic. But for example, what about CO2? It does not have any hydrogens. So CO2, it's not organic, but this one is inorganic. Okay, so with the organic molecules, the types we're going to look at: carbohydrates, proteins, lipids, and nucleic acids. We're going to start with the carbohydrates. Carbohydrates are really just sugars. Okay, so they have carbons, hydrogens, and oxygens. Now what I want you to know: monosaccharide just refers to a simple sugar, just the one single sugar. So know that glucose is an example of monosaccharide. Okay, so it's just one molecule. But a disaccharide just means when more than one sugar molecules have come together. So if it is more than one disaccharide, and so here's some examples. But then I want you to know for the polysaccharides, this is more than one sugar molecule that have attached to each other. And for this, I want you to know that these are the different types. There's starch, glycogen, and cellulose. All three of these are made up of many glucose molecules. It's just that they're arranged a little bit different. Animal cells store glucose as glycogen. Plant cells store glucose as starch. And then cellulose is more for, adds more strength to the plant material. So that's also for plants. Okay, so again, what I want you to know: monosaccharide, glucose is an example. And then for the polysaccharides, know that there's starch, glycogen, and cellulose. And know that glycogen is what's in our cells. We don't have starch in our cells. That's only for plant cells. So this is just showing the formula for glucose. Actually, on the left side, this is showing just the structural formula. When glucose is in water, when you dissolve it, it turns into this ring form. Okay, and then this third picture, they're not, they didn't write down the carbons and hydrogens in this one, but it's basically the same thing. And this is just showing how molecules can come together. So monosaccharide, again, just know that it's just one sugar, for example, just one glucose, or it could just be one fructose. Disaccharide means more than one molecule have come together. And a polysaccharide, a lot of sugar molecules have attached to each other. Okay, and then next, lipids. Lipids are oils and fats. Now, lipids are insoluble in water, meaning that they don't want to mix with water. The other word for this is hydrophobic. All lipids are hydrophobic. But on the other hand, if you can mix something in water, for example, sugar or salt, that's hydrophilic. Okay, so lipids, the different types of lipids, we're going to look at triglycerides or fats and oils. Then there's also phospholipids and steroids. Okay, so we're going to start with the triglycerides. Triglycerides give our cells energy, give our body energy. They contain actually more energy than carbohydrates. Okay, so what I want you to know for triglycerides is that they contain glycerol and 3 fatty acids. Okay, so what does that mean? Let's look on the next slide. So here, this is a triglyceride. This orange part here is one glycerol molecule. Okay, so, and you do not have to memorize any of this. You don't have to know the exact structural formula or anything, but know that there's a glycerol molecule, and then these pink, looks like rose here, those are the fatty acids. So there are 3 fatty acids total. So fatty acids are just like a bunch of carbons and hydrogens attached to each other. Now, let me show you this. Some triglycerides may have a double bond in the fatty acid. So here you can see the 2 lines. This refers to a double bond. So this means that more electrons were shared between these 2 carbons. That's all it just really refers to. Having a double bond gives the fatty acid it's kink. So it makes it bend a little bit. Okay, but again, the important part, triglycerides are made of glycerol and 3 fatty acids. Saturated versus unsaturated, so this is where that double bond comes in. Saturated fats, no double bonds, only single bonds. But unsaturated fats have double bonds. Okay, so you can see here, this one is saturated, no double bonds. But this is the unsaturated, we're seeing the double bonds here. Having these double bonds in the fatty acid makes that triglyceride more liquid. And so the unsaturated fats are oils, so plant products, but saturated fats, think like, let me write it down this way, think butter, lard, so animal products are the saturated ones. Okay, next one, phospholipids. So again, these are still lipids. Know the difference between phospholipids and triglycerides. Phospholipids have a glycerol, just like triglycerides, but only 2 fatty acids, not 3. And they also have this phosphate group. What this phosphate group is, let me show you on this picture here. This here, the phosphate region, so it's basically a phosphorus attached to some oxygens, and it's also attached to some carbons, hydrogens over there. Okay, so it's just a phosphorus atom with some things attached. But this difference makes phospholipids different because this phosphate side is actually okay with water. It's okay if it touches water. Okay, no, the phosphate is hydrophilic, but then the fatty acids are still hydrophobic. Okay, so what they did here, they made a picture of a phospholipid. This part is the phosphate side. This part is okay with water, but then those two parts here look like tails, like little legs. Those are the fatty acid parts, and they still don't like water. But so we're going to see that phospholipids, because of this property, they make up our cell membranes. We'll see that on the next chapter. Steroids. Steroids are just many rings of carbons. Know that steroids help to make cholesterol. I know we think of cholesterol as being bad, but actually cholesterol is very important for our cell membranes. It helps to keep our cell membrane stable. Cholesterol is steroid and also sex hormones. So what I mean by that is estrogen, testosterone are also steroids. Okay, next we can move on to proteins. Proteins are used to make up our cells, but also give us a little bit of energy. A lot of proteins are hormones, receptors, enzymes, antibodies. Okay, so proteins are made up of amino acids. So each amino acid is one molecule. So when many of these amino acid molecules come together, you get a protein. We have 20 different amino acid molecules though. So there are the different kinds. So what we're seeing at the bottom, if we look at these pictures, glycine is one example of an amino acid, alanine is an example, valine. If you look here, they have a lot of things in common, 3 things in common. There's a carbon in the center, then there's a hydrogen at the top, there's the H3N to the left, and the COO to the right. So if you look at all of these, that's the same. But what is different? What makes these amino acids different is this bottom part. Okay, this bottom part is called an R group. And so the R group can be just one atom or many atoms that have come together as molecules. So glycine is always going to have this hydrogen at the bottom, or in this R group. But then alanine has CH3, valine, CH3. Of course, you don't have to memorize these. But so this is what's making them different. So the R group is like an ID tag for the amino acids. And so then the amino acids come together. What this is trying to show is here's one amino acid, here's another amino acid. And with the condensation reaction, or anabolism, they get together. The same thing as the synthesis reaction. Oh, actually, what I want to know here, peptide bonds is the bond between amino acids when they attach to each other. So proteins are made up of many amino acids. So if you look at this top picture, here you see many different amino acids have attached to each other. So just the chain of many amino acids is called a primary structure. So the primary here, many amino acids. Secondary structure, what happens in proteins is that sometimes you get these weird curly shapes, pleated shapes. So if these shapes arise, and these are actually because of hydrogen bonds, these shapes are called secondary shapes. So secondary structure, pleated or twisted shapes. The pleated one is actually called the beta pleated sheets. The twisted one is called the alpha helix. But know that those are shapes that can appear. And so then you're seeing here one blob, so one protein. This is called the tertiary. So a tertiary just refers to the amino acids that made up this protein. So you're seeing here how there's the secondary structure, the secondary shape. Let's see, is there a pleated one? I guess this is supposed to be the pleated one here. But so one complete protein is tertiary. If more than one blob, I know blob is not a very scientific word, but if more than one subtype of the tertiary have come together, then it's a quaternary. So this is for large proteins. So for example, our hemoglobin protein that carries oxygen in our red blood cells, it's made up of more than one part. Okay, the next nucleic acids, DNA versus RNA. DNA carries the genetic code and RNA helps in making the proteins. Now, both DNA and RNA are made up of nucleotides. Nucleotides is just one molecule. So here, this picture is showing a nucleotide. Nucleotide has a phosphate group. It has a sugar. And it contains another molecule called a base. Now, the bases can be different. And the bases are made up of a lot of nitrogens, hydrogens that have come together. Here are the different types of bases. And we're going to see how the DNA versus RNA have different combinations of these bases. So in this picture, we're seeing RNA here. This is DNA. RNA has just the one line here. This gray line, this is representing sugars and phosphates. Sugars and phosphates make up the rung, if you want to think of it this way. And then these color parts, these are the bases. And so you can see here, again, you don't need to memorize what these bases are, but you can see how cytosine, this is the formula for cytosine, the guanine, this is what it looks like, adenine, uracil. Okay, so what is important is that RNA is single-stranded, and it contains these bases, and DNA is double-stranded. And so it has the two lines of sugars and phosphates, so that's the gray part, the long gray parts, and the bases are in the center, and in DNA the bases are attached to each other because there are the two parts. Okay, so important stuff here. I'm going to put a big star here. Know the difference between DNA and RNA. Know that DNA sugar is deoxyribose. It's double-stranded, double helix. And know that its bases is A, T, C, G. Okay, I'm not going to make you guys write down the names of the bases, but definitely know the first letters at least. And then RNA, its sugar is ribose. It's single-stranded. And this one, instead of having thymine, it has the uracil base. So it goes AUCG. Okay, so that's the difference.